It's not terribly professional, but it'll work.
I put in the wrong class all
Well, that shows how much you come to class. Got it? Okay. Okay, well, the first part of class, I want to just go through some problems again, and then I'll talk a little bit about the final, and we'll do evaluations. I will have your extra credit assignment back to you by the time you come in for the final, which I believe is Tuesday, 10 to 12 next week. So you'll have those. Most people are doing really well on it so far, so I'm real pleased about that. It suggests that sort of this is making a bit more sense than perhaps the earlier material did. I hope it is. The final exam has a good, 20 some odd questions just on problems. So you need to know how to do these. And that's why we've been doing so much practice with them. All right, so here's a pedigree I just sort of threw up out of my head about a half an hour ago. The first thing we might ask about this pedigree is, what is the mode of inheritance? And you probably looked at it as you came in and sort of puzzled it over. Again, you'd follow your sort of standard rules for figuring out these sorts of things. You'd look at it first and ask, is it dominant or recessive? Does it appear in every generation or does it not? Right? Clearly it appears in every generation, so it must be dominant. Then the next question to answer is whether it's X-linked or autosomal. To answer that question, you're better off looking at the progeny of affected males if you have any. That's a better thing to look at. You can't tell X-linked versus autosomal dominant if you look at affected female offspring because they look like the same thing. So you look at the affected male and you see that this affected male produces all affected daughters and no affected sons. All right? That's typically X-linked. All right, so it's an X-linked dominant. All right, so we've got our X-linked dominant. What are the next question we might ask is, what are the genotypes of all the individuals in this pedigree? Now, to do this, you need to know that typically if a female has an X-linked dominant trait, she's heterozygous. Yes, there's a question.
If it's X-linked dominant, why would there be a female without a second generation?
Because, here? Because this female has two X's, right? And if it's X-linked dominant, only one of those X's need carry the trait. Yes, exactly. Okay. I love that. I saw that little light bulb go on. That's great. Pretty cool. Okay, so you typically figure that these affected females are going to be heterozygous for the trait. But in fact, we know that this female is heterozygous because she produces an unaffected daughter. So she must be heterozygous. So if I let, I'm going to use the symbols X big A and X little a to illustrate both the X chromosomes and the alleles that they carry, either the big A or little a allele, the dominant or recessive allele. And then I'll just use a Y to indicate the Y chromosome. Okay, so we can indicate the genotypes of pretty much everybody in this pedigree. We know that this individual is heterozygous because she produces an unaffected daughter. So we can say she's X big A, X little a. This male would be X little a Y. Right? He does not show that dominant trait, therefore he must be carrying the recessive allele. In the next generation, this female over here on the far right is X little a, X little a. This male here is X little a Y, because he does not show the trait. This female must be heterozygous. She picked up this dominant allele from her mother, but she picked up the recessive allele from her father. So she's X big A, X little a. The male here is X big A, Y. And this female over here on the far left must be X little a, X little a. Okay, so now we can do the next generation. This male must be X little a, Y. He does not show the trait. This affected female picked up the dominant allele from her mother, but the recessive allele from her father, so she's heterozygous. This male that's unaffected must be X little a, Y, and then these two females are both X big A, X little a. All right, so it is possible in pedigrees much of the time to actually be able to indicate all of the genotypes of the individuals, particularly if you're dealing with a dominant trait. Might not be so easy with a recessive trait because somebody who's unaffected could be heterozygous. They could be a carrier for that trait. So sometimes it's not as easy. All right, so we indicated the genotypes of all these individuals in this pedigree. We then might ask, in the mating indicated by the question mark, this consanguineous mating right here, right? This is a first cousin mating. Okay? What is the probability of some different genotypes? What's the probability of an affected child? To do this, to answer the question, you could just do a Punnett square and figure it out. It's fairly easy to do that. We know the genotypes of the parents, so we could use those to find the genotypes of the offspring. If we did a Punnett square like that, okay, we haven't asked about the sex of the individuals. So we're just looking at affected individuals. We could have affected males or affected females. And we get affected offspring about half the time out of this Punnett square. So the probability of having an affected child is 1/2. We might ask what the probability of an unaffected child is. And you see by this Punnett square that we did that that is also 1/2. A lot of times on problems like this, you'll see situations where they show you the pedigree, you have to figure out the mode of inheritance, and they'll ask you questions about the probabilities of having particular phenotypes of offspring from different crosses in the pedigree. Okay? So they sort of skip these intermediate steps. I didn't want to do that. I wanted you to see all the steps in between so you know conceptually what to do while you're trying to solve these kinds of problems. So if you see a problem that gives you a pedigree and all it asks is probabilities, okay, well, mentally and perhaps on paper as well, you might want to go through these other steps. Figure out the mode of inheritance first. That's going to be critical. Then you might want to figure out the genotypes of maybe not all the individuals, but at least the ones that are relevant to the probabilities you're trying to figure out. And then you can answer the question really easily. Okay, any questions on that pedigree? All right, so we've gone a step further. We've gone from just looking at a pedigree and saying what's the mode of inheritance to really asking some questions about that pedigree. All right, what are the consequences of this particular trait and how it's inherited? Okay, let's suppose that we are looking at, here's another example. Let's suppose we're looking at the inheritance of black versus gray fur in cats. And our first piece of information is this, that several matings between different pairs of black cats consistently produced all black offspring. Okay, so the question is, can we conclude that gray is recessive? Okay, let's say that we went out into the population, we got 100 black cats. We did 100 different matings randomly between black cats in that population. Okay, we didn't take any of the gray cats in the population. And we see that they consistently produce all black offspring. Okay, if you think carefully about this.
Black could be recessive.
Absolutely, exactly. Black could be recessive, right? Which would imply that, in fact, these black cats are all homozygous. They're all true breeding. Right. So as a result, you know, they would not segregate the gray trait simply because they don't carry the alleles for the gray trait. So the answer then is no. Black could be recessive. Okay. And or the black cats are all homozygous. I mean, it could be that black is dominant, but these black cats that we're using are all homozygous dominant for the trait. So they don't segregate it. Well, that's an interesting question. The question is, what's the possibility of that actually happening? The answer, as we'll see next term, depends on the frequency of the alleles in the population. Right? If 1 in 1,000 individuals is gray, then the probability of picking up 2 cats that are homozygous for black is actually pretty good. Yeah. Right? It all depends on how rare those alleles are in the population. Okay, let's say that we take a black cat, cross it with a gray cat, we produce an F1 that contains black cats, and then we cross the F1 black cats, and they occasionally produce a gray kitten. All right, so can we know which trait is dominant and which trait is recessive from those data? I didn't tell you, did I? No. Yeah, you can't assume that. All you know is that when you take black cats and gray cats, you get F1s that contain black cats. You don't know if they contain gray cats or not. But when you cross the F1 black cat, you occasionally produce a gray kitten. Actually, from these data, it is fairly clear which one's dominant and which one's recessive. Okay? You can tell. The reason is because you've got cats that are black that segregate the gray trait, even though you don't see it in those black cats. They're carriers. They're heterozygous. The trait that is expressed is black. The trait that's masked is gray. Therefore, black is dominant and gray is recessive. You know that from this cross. The other piece of information that helps you along there is that it says, well, they occasionally produce a gray kitten. You expect, if these cats are heterozygous, you expect that that's occurring 1/4 of the time. Right? 25% of the offspring, on average, ought to be gray. Right? That's if there's nothing else funny going on. You remember I talked about pleiotropy, where one gene has many effects. Perhaps this has an effect in that it causes embryos to not develop to term, that are gray, so you don't see as many grays. Right? It's the same idea with Turner syndrome. Turner syndrome is far more common in embryos than it is in live births in humans. It's because the embryos tend to die at a certain fetal stage, and you don't see them in the offspring. So if you were just counting offspring that were Turner syndrome, you'd see it far less often than it actually occurs. Okay, so from these data, you can in fact determine which trait is dominant and which trait is recessive. But from these data up here, you cannot. Okay, here's another fun one. Let's suppose that the average pea pod has 7 peas in it. And let's suppose that we cross F1s from a round times wrinkled parental cross, and we'll indicate that those parents were true breeding. What's the probability that an F2 pea pod has all round peas? Okay, so this is a basic monohybrid cross. We're not making anything difficult here. We're taking true breeding round, crossing it with true breeding wrinkled. We get an F1 where everybody's round. We cross those F1s to each other. We get F2 plants, which are going to have seeds that are either round or wrinkled, depending on how this is going to segregate. Okay? So what's the probability of having a pea pod with all 7 peas being round? It's not as difficult as you might think. To answer this question, the first thing you have to figure out is what's the probability of a round versus the probability of a wrinkled. Okay? And to do that, if you wanted to, you could do a little Punnett square if you didn't remember the monohybrid crosses. I'm just going to use the symbols big A and little a, sort of generically. But you should remember it by now that when you do monohybrid crosses, then the dominant phenotype shows up 3/4 of the time, and the recessive phenotype only shows up 1/4 of the time. So when the probability of a round is 3/4 and the probability of a wrinkled is 1/4. Okay? Now each of these P's in these P pods are independent events. Okay, because of the way the segregation occurs. So if you have 7 P's and you want them all to be round, then the probability of that is just all of the individual probabilities multiplied together. You're using the AND rule. Right? So it's the probability of having a round times the probability of the next one being round times the probability of the next one being round. And since they're all independent events, you just multiply all those probabilities. So it's just 3/4 to the 7th power. 3/4 times itself 7 times for each of those P's. What does that come out to? Well, let me look. Comes out to about 13.5%. If you actually multiply it through. Now, of course, if you saw something like this on an exam, I would not make you multiply it all the way through. You'd see something like this answer, where it was like 3/4 to the 7th or 3/4 to the 5th or whatever. Okay? Would not make you multiply it through. I mean, this is entirely analogous to asking the question, let's suppose a family where the parents are normal but heterozygous have 7 children. What's the probability that all 7 are normal? About 13.5%. Okay? It's the same question. Right? Think of peas as plants' children. And it won't go wrong. Right? Okay, let's suppose that we catch a black stallion from the wild and cross it to several purebred sorrel mares. Okay? Sorrel is sort of a red color, isn't it? Yeah. All right, so we catch black stallions from the wild, cross to several purebred sorrel mares, and out of those crosses we produce 29 sorrel and 33 black foals, offspring. 29 sorrel, 33 black offspring. Which trait is recessive? First, ask yourself what kind of ratio this is. That's roughly a 1-to-1 ratio. That may help you determine which trait is recessive. Okay, so which trait is recessive? The sorrel, yeah, has to be. The reason is because these sorrel mares are purebred. You cross them with the black stallion, and you get segregation of these traits, these two traits, which means that this black stallion must be heterozygous for black versus sorrel. So this is nothing more than a test cross, where the black stallion, we could say, is big B, little b. The sorrel are all little b, little b. So when you cross those two together, you expect 1/2 big B, little b's and 1/2 little b's out of that. Well, the big B, little b's end up being black. The little b, little b's are sorrels. Okay? Y'all are going to smoke this exam. This is going to be like second nature to you. This is good. I mean, there's no problems on this exam that you haven't already seen in class. I mean, the same concepts, so shouldn't be difficult for you. All right. As far as the final, oh yes, question.
If you don't say that they're X-linked, can we assume that they're not on the exam?
Yes, you can. Yeah. Besides, with this particular situation, you wouldn't expect this kind of a situation to occur if it was X-linked. You'd expect one sex or the other to predominate in terms of one phenotype or another. Typically, if a problem does not indicate males and females, you don't have to worry about the linkage. Also, typically a problem, if it wants you to determine the linkage, it will ask you to determine whether it's sex-linked or autosomal. Otherwise, typically they'll tell you they're autosomal. Or you can deduce that from the ratios. Okay? Because the ratios are a little bit different between X-linked and autosomal traits. Okay, any other questions? Okay, final exam. Do not bother reading the chapters again. It will be a huge waste of your time. Here's the two things you want to do as far as the old stuff goes. You want to look over your notes a couple of times just to make sure you remember all the concepts. Look over your exams pretty intently. The reason is because the major concepts were tested on those exams, and so as you go through those exams and refresh yourself on those answers to each question, what you're doing is you're pulling out all of that information, okay, and studying it as you study those questions. All right, so the old exams, pretty important. Notes, look over them once or twice just to make sure that you haven't forgotten anything major. Yeah?
Are all the exams the same, it's just in different order? Like, are the questions the same?
Yes, in all the different forms of the exams, the questions have been the same. It's just a different order. The reason I do it that way is because potentially exam A might have more difficult questions than exam B. And that's not fair to the people who got exam A versus exam B, so it's the same questions, different order. Right. Okay. Just so you know, they're scrambled completely. Okay, they're not page by page, so you can't, look over at your buddy and say, okay, what page are you on? I'll flip to that page. It doesn't work that way. The scrambling is complete. Okay, so old exams, notes once or twice. Now, as far as the new stuff goes, study that like you studied for the first two exams. All right, concentrate on that. This is 50% of the exam. All right, so this is 100 points. This review stuff is 100 points. All right, so the final is worth twice as much as a regular exam. With the new stuff, there are, as I said, there's approximately, I don't remember, 25 to 30 questions on some problems, genetics problems, and there's a fair number of questions on the stages of meiosis and mitosis, so make sure you know those stages and what happens in each of those stages.
How many questions will that be?
There's 100 questions, each worth 2 points. Yeah. I toyed with the idea of only having 50 questions and having them worth twice as much, but the reason I didn't do that is if you miss 3 questions, that's 12 points, for example. Whereas if they're worth 2 points, you can miss 6 questions for 12 points. So it actually works out a little bit better for people if the questions are only worth 2 points, because that way if you don't remember something, you don't get dinged as much. It's not as critical to the grade, the final grade for the final exam. But yeah, each one is worth 2 points, so there'll be 100 questions. So the meiosis and mitosis, know those stages, know what happens in each of them. When you're dealing with chromosomes, remember, count centromeres to count chromosomes. All right? Don't count chromatids. Count centromeres. And always remember in meiosis, you have homologous chromosomes that have duplicated their chromatids. They pair. Then in the first meiotic division, the tetrad separates, and then the second division, you separate chromatids. Okay? Right? You just do the finger thingy. Can't think of anything else that will really help you along except to make sure that you study the material and be ready for it. Are there any other questions that are needing to be asked? Okay, the next thing I want to do is evaluations. So you'll need, as I asked on Wednesday, I asked you to bring a number 2 pencil. If you don't have a number 2 pencil, a black pen will